today we have Professor GIAN Course from Department of Nutrition Biology, Central University, Haryana. And his work is very much related with the probiotics and its application in gut microbiota. And Professor GIAN Course is working on artificial science system also in his laboratory. So his area of expertise and his views over it, how probiotics can be applied in modulation of microbiome in the system is very useful for you all. So this is the reason I requested Professor GIAN to deliver his views on probiotic application in the animal and human, how it can be optimized in the artificial system. Because sometimes it is very difficult to work with animals and you don't get optimized result. And if you optimize the things outside the system and get the conditions first verified, optimized, and then you can use the animal system for its application. So sir, we are waiting few minutes for participants to join. Actually, so the session starts at 11, but they take like 5 to 10 minutes to join. So in few minutes we will start.
Okay, sure.
Yes, sir.
Okay.
Dear participants, if you have anything query related with the previous lecture that we can discuss, meanwhile in a 5 minute, and whatever Professor Chikinda delivered today was mostly on the gastrointestinal tract and its probiotic modulation using bacteriocin producing bacteria and others, and that may be helpful for correcting several diseases. So there may be use of several genomics platform also we discussed last time, and the lecture was very fruitful, and we found that this very link with the today's tutorial. This is the reason we have connected with Professor Goel to have some idea on the gastrointestinal tracts, microbiota and their modulation. So this is really a tutorial based on the previous lecture we are having, and if you have any query, anything related with the last lecture, you can discuss. Otherwise, I will have some short announcement to you that all the participants are advised to join because our lectures are video recorded. Your attendance are going directly to the IIT Kharagpur. They join from here. So you cannot hide. Number one. Number two, you have to join with your name. Don't change that. The name you have registered with us, kindly use the same name to join with our portal. Otherwise, it will be difficult to calculate your attendance and give certificate. After the lecture, as you are all aware that we are having an examination on 25th of this month, tomorrow. On 25th, there will be exam for 1 hour, 5:30 to 6:30. In the morning, we will have tutorial in the same schedule, but in the evening, Dr. Chikinda's lecture is not there. At his lecture, we will have 1 hour examination. The pattern of examination will be based on the assignment. You have to submit an assignment within 1 hour. So you have to be on portal here and write on page for 1 hour on a particular topic which will be given at that time. And the topic will be based on the lectures delivered by Professor Chikindas and the tutorials. So be careful. I am posting the lecture of Dr. Chikindas too on your mail also. So for your reference, you can again go through his lecture and prepare accordingly. So yesterday and today's lecture when complete, I will send it to you again that you can have. And I thought that it will be a kind of material to you which for future reference you can go through the handout we are giving to you. So for any doubt or clarity you can ask me, otherwise we'll start the tutorial section. So Dr. Gunjan, you are ready from your side or you need time?
Yes, no problem. If the participants are there, I'm ready.
Okay, so I formally welcome Professor Gunjan Goyal from Department of Food Nutrition Biology, Central University, Haryana, and on the behalf of this organizing committee, we are heartily thankful to him that he accepted our invitation to deliver his ideas on the GIT. Professor Gunjan Goyal, over to you.
Okay, thank you, Dr. Tiwari, and very good morning to all of you. And I thanks Dr. Tiwari as a GIAN program organizer for me on this session. And as he already said that all the GIAN program was mainly on probiotics and how we are using the probiotics in our daily routine and what are the applicable part of the probiotics. So I'm also touching the same part, but my laboratory, we are mainly focusing on how to simulate this gut microbiota in the laboratory conditions. Because I from the diverse backgrounds, some might be of zoology, maybe botany, maybe microbiology, maybe biotechnology participants. So to have a brief on this gut microbiota, because nowadays everything is linked to the gut microbiota, that how our physique, our mental status, our digestion, our physical activities, all are related to the gut microbiota. What are the different kind of microbes we are inhabiting, how they are behaving in our gut, whether they are surviving, whether they are dealing with pathogens, so nowadays everything is with the gut microbiota. So normally, whatever the work we are dealing with the probiotics or maybe any functional food material, we generally use animal models. But with the animal models, like most of the institutes, they don't have the facility of animal house, and it is very difficult to get the approvals from the animal ethical committee. You have to decide how many mice you will be taking. And some of the institutes, they are not permitted to inbreed the mouse. So most of the times it is very difficult to handle the in vivo studies. So what we have seen that maybe we can, like for the gut microbiota studies, we can simulate those conditions in the lab. And although it is also very difficult to simulate the exact conditions of the gut GIT tract in the laboratory conditions, however, there are ways by which we can simulate these conditions in the laboratory condition. I don't say that it is exactly mimicking the in vivo condition, but somehow we are making a replicate of our GI tract in the laboratory. So that's what we are going to discuss today. So let me, I have divided this tutorial in two parts. I have a part of this PowerPoint presentation, and then I will just switch to videos of our laboratories. So let me show you that presentation. Is my screen visible to you?
Yes, sir.
Okay. So we will be dealing in this tutorial. We will be dealing the how to set up the simulated gut environment in the laboratory conditions. So, like I said, that it's more most of the time now. Most of the studies have been dealing with the gut microbiota studies. How different food ingredients, how probiotics, how different conditions they affect our gut microbiota, and that is because mainly the rise in the publications of the gut microbiota studies. Although in India we are having few laboratories which are working on gut microbiota, but it is not as vast studies have been conducted in India as the Western countries. So, like I said, that we are going to simulate all the conditions in the laboratory conditions. So let me have let us have a look on this our digestion compartments. Like all of us have studied that we are having four different major compartments. That is the stomach. Starting from the mouth, then the stomach, then the small intestine, and the colon region. So I have highlighted this in yellow. The transit time, that how much time any food material, any drug, or any particular component that stays in our that compartment, that is known as the transit time. So like in mouth, whatever we are having, whatever we are eating, it undergoes different mechanical things like chewing, mixing, matrix disruption, phase transition, structural changes, and there may be have action of amylase enzymes. So normally the food stays in our mouth for 10 seconds to 2 minutes. And if you see the bacterial count in this mouth condition, we are having around 10 to about 11 CFU per ml of the content of the mouth. Passing on from mouth, then the food goes to the stomach, where we have a transit time of 15 minutes to 3 hours, and the very less bacterial count, that is 4 10 to about 3 CFU per ml. Moving on to stomach to the small intestine. In small intestine, normally we have the digestion process, where we have a major macromolecular degradation, mixing, dilation, absorption of the nutrients, and we have secretion of enzymes like proteases, lipases, amylases. We have bile and sodium bicarbonate. Here the food stays for around 2 to 5 hours, and the bacterial load is generally it is around 10 to about 4 to 6 CFU per ml. Then from the small intestine, the food passes to the colon region. That is a major, you can say the fermenter or the reactor, or you can say the major reactor conditions where we are having a pH of 5 to 7, and the food stays in this colon region is around 20 to 24 hours. And we are having thousands of microbial species which are inhabiting our gut, and the general count of bacteria is around 10 to about 12 to 13 per ml. So in my slides, whenever or in my talk, wherever you have some confusion, you just stop me wherever you want, and we can discuss. So this is the overall picture of the digestion compartments of mouth, stomach, small intestine, and the colon. And normally, going through this human gut system, we have two kind of studies, mainly the physiological studies and the biological studies. And there are the different models which we can go for these kind of studies. First is the in vitro gut fermentation model. Then the human intestinal cell models, like you can have like different cell lines. Then we have the in vivo animal models, and then also we can go for the in vivo human studies. But all of these studies, whether it may be in vitro or in vivo, normally we study the complex microbial host interactions which govern the function of our gut microbiota. And in the foods, normally there are 5 B's which are more prevalent in this food digestion. These are the bioaccessibility, how much particular or functional ingredient is released from the food, how much of that is absorbed, that is called bioavailability, how much of that is converted to the active fraction, that is bioconversion, and how much effective is that particular molecule, that is bioefficacy. And bioequivalence is a term which is more related to the pharma drugs. And during these studies, we can study the gut microbiota through the various modes like metagenomics, proteomics, transcriptomics, and the metabolomics. So various in vitro and in vivo methods are already described in the literature. With the in vivo methods, normally like most of the laboratories, they have more concern with the ethical concerns, and we have to follow the severe protocols of the ethical committee or the human ethical committees. Whereas the in vitro methods, the major problem is with the contaminants. Although they are easy to conduct, but the major problem is with the contaminants. So usually In literature, we have the batch culture systems, continuous systems, and the microbial host interaction models. In batch culture, we can assume just like you can assume a bioreactor. Like some of you might have run some bioreactors, small bioreactors, maybe 1 liter, maybe 2 liters. So it is that kind of a study that you add the inoculum, you add the feed ingredients, everything there. You set up the fermentation, you conduct it for like say 24 to 72 hours, and then you analyze the fermentation. So it is a kind of a batch culture, like putting everything together, let the fermentation finish, and then you analyze the fermentation. Then we have single stage continuous model or the multiple stage continuous models. These are the continuous systems like which mimic normally the human digestion pattern. Like we eat the meals 3 times a day, so we are metabolizing that meals and then it is excreting also. So in the same way, we are conducting the in vitro studies where we are feeding the reactors and also we are extracting the waste of the reactors. So these studies are called as the continuous systems models. And nowadays, more advanced techniques are available where we can actually study how the microbiota is interacting with the host. So normally in the lab, first, whenever we start with the simulated experiments, we start with the batch fermentation. Like let us say that we have to analyze that how much of particular that bioactive component, let's say polyphenols, how much they are active in the gut microbiota. But as you all know that polyphenols, they may have like antimicrobial action also if they are taken in the very high doses. So normally in the batch cultures, we optimize the reaction conditions and also we optimize the dose concentration of any active component. So these I said that these are very small kind of reactor. We use the serum bottles to conduct these kind of experiments and where we optimize the dose of a particular component. So what are the advantages of the batch culture? These are very fast, they are cheaper, they are easy to operate, they are reproducible. You can make several replicates also, 3 replicates in a day and 3 times perform. Then you have 9 replicates within a single set of the test compound. And they offer like very reduced cost, they are very efficient systems. But only disadvantage is that we are not actually mimicking the simulated gut environment because it is having the very short culture periods. Then are the colonic model systems. Like some of the systems, this is one of the picture from 3-stage colonic model system where we simulate only the colon part. That is, you can see there are 3 vessels, vessel 1, 2, and 3. And we maintain a pH of 5.5, 6.2, and 6.8 in all of these 3 vessels, and we simulate the colonic fermentation in these vessels. But this system, it does not have the stomach and the small intestine part in the system. Later on, moving on to the most widely accepted model system, that is the GIAN, that is simulator of human intestinal microbial system, that is developed in Belgium, University of Ghent, and that was in 1993, and still they are using the system. Now they have developed like now they have more advanced system with where we can study the human microbe interaction also. So here also they have the last three part are the like the colon region, that is the ascending colon, transverse colon, and descending colon. Along with it, it is also having the stomach. They have simulated the stomach and the small intestine conditions. And it is a comparison of all the systems which are available worldwide on the three reaction model systems. So what happens during the batch fermentation systems? Like we have studied different food juices and how they have been performing in the batch system. So to simulate, like in the laboratory condition, whenever you produce an enzyme, you just have an inoculum. But in this case, because we are going to simulate the human gut microbiota, we take fecal sample as an inoculum. And that fecal sample is to be collected from the healthy volunteers. That is must. And these volunteers, they have not undergone any antibiotic treatment. They don't have to get any smoking or alcohol. So we make you can say a questionnaire. Based on that, then we take the human samples from these healthy volunteers. Then you mix this human fecal samples, and normally we take 5 to 10 human volunteers for the feces, and then we mix them uniformly, and then add in the batch fermentation. And after the completion of fermentation, we can analyze the fermentation for different metabolomic studies as well as the microbial communities. So this is the composition which we use in the batch fermentation, that is mostly mimicking our colon content. And this is the continuous system where we have couple of series of connected reactors. So you can see there's the glass reactors, double jacketed vessels starting from the stomach, then the small intestine and the large intestine part. So all of these connectors, all of these reactors, they are interconnected with the feeding tubes. Like in our digestion system also, like food goes from mouth to the stomach, then the small intestine, then to the large intestine. So we have connected these all reactors with a very fine tubing, and these tubings are connected with the peristaltic pumps. So these pumps move the feed from one compartment to the other, second to the third, and third to the fourth, and fourth to the fifth, and finally to the waste. In stomach, normally, like say we have an acidic pH, so we have created a medium which we have the acidic medium. Then in the small intestine, we added the pepsin and the pancreatic enzymes to simulate the small intestine condition. And once the liquid flow from small intestine to the colon, we add our inoculum over here. So there we start the fermentation things in the colon reactors. And to maintain the pH of these colon reactors, these reactors are connected with the pH controllers, which exactly maintain the pH in these kind of reactors. So this is the basic design of a fermenter, which we have customized designs. So it is a double jacket reactor. The advantage of double jacket reactor that you can move the water at 37 degrees, so that it is not like every time that it is like if there is electricity failure, then you can have a drop in the temperature. So it completely maintains the pH temperature of 37 degrees. Then these are the outlet and the inlet of the water. And at the port, at the top of the lid, you can see there are 7 to 8 ports where we have an inlet of the feed, outlet of the feed. We have to flush it with the nitrogen so that to maintain the aerobic conditions in the reactor. Then we have a pH controller ports where we feed the acid and the base from the different parts. And in the middle, we have the pH probe, which actually gives you the exact pH in the reactor. This is like I said, that the transit time is more important. So we have simulated these conditions with a different residence time. Like in the stomach and the intestine, we have decided a residence time of 4 hours. And the ascending transverse and the descending colon, we have put up the residence time of 20, 32, and 24 hours. And we have to maintain the flow rate so that it actually maintains the similar flow rate throughout the study. And we have to maintain a pH of 5.6 to 5.9, then 6 and 6.6 in the colon regions. This is the composition of feed, which is mimicking our what are the contents in our gut, and this is the composition of the pancreatic juice. So normally we run this system for continuous 21 days. So microbes are more sensitive to the environmental conditions. So in starting first 7 days, we go for the stabilization period. So when we add the fecal sample in our continuous system, it has to undergo the stabilization. And normally we have seen that up to 4 to 5 days, the gut microbial population is stabilized. And this we check with the plate count method and the production of short chain fatty acid. That is a major metabolic activity of the gut microbes. Then we go for the treatment period, whatever the things you want to test. After the stabilization, you add that component in that continuous system, and then you can test what kind of things changes you can observe in this gut microbial composition. And finally, after the treatment period, we have a washout period. We remove the treatment and we see actually that whatever the treatment you have given, whether it has a stable impact on the gut microbial population. And during this whole 21 studies, we have to conduct the microbial analysis, we have to conduct the short chain fatty acid production, we have to conduct the metabolomic assays, and then we can go for the NG study for the microbial community. So this was all about that what kind of experimental setup we can perform in this gut microbial studies. Now I will share a video. Are you able to see the video?
Yes, sir.
Okay. So this is a setup of the batch fermentation, how we simulate the batch conditions. So these are the small reactor vessels, which we normally use. It is a capacity of 125 ml, and we fill it with the content with the 80 ml. So the feed which I have shown, that we add that feed in these particular bottles. So here in these bottles, we have added our substrate, and beforehand, like maybe 1 milligram, 20 milligram, or different doses of the substrate in each of the bottles. Then This is the artificial juice where we have the bile and several components which are mimicking our content of the gut microbiota, gut microbial content. Normally, it has a capacity of 125 ml and we fill it with the 80 ml. And you can see that it is around a pinkish color of this content. That is because of the pH indicator, that is resveratrol. Resveratrol, it indicates whether you have an aerobic condition or the anaerobic condition. So we fill these bottles with the 80 ml of content of this content of the intestine. And when you subject this with the autoclaving, and autoclaving, normally this resveratrol loses its color, which indicates that we have a proper anaerobiasis in the bottles. So all these bottles, they are with the different different treatments, different doses of the bioactive components. So after filling these bottles with this content, then we have this system of a gassing manifold. It has copper wire at the top, and this copper wire is heated. So normally for anaerobiasis, we inject with the hydrogen or with the carbon dioxide. The market available carbon dioxide and hydrogen, they are not completely, you can say, free of oxygen. There might be some traces of oxygen. So when we subject the gas to this heated copper wire, it absorbs the traces of oxygen. So this gassing manifold, we make ensure that we are flushing oxygen free the carbon dioxide. And you can see there are the three ports, that is one with the hydrogen, nitrogen, and the carbon dioxide. And when you flush with the gas, and there is a pressure bar which indicates that the gas is being flushed in the bottles. So there are the different ports through which we can flush the bottles with the different gas composition. Normally we do with the carbon dioxide, but we can manipulate the things. We can you can say mix the gases in the proportion that you can mix like 70% of carbon dioxide, 20% of hydrogen, and then 10% of nitrogen. So it depends what kind of experiment you want to set up. Normally with the gut simulation experiment, we just flush it with the oxygen-free carbon dioxide. So when the copper wire is heated, we turn off the heating and then we start flushing the gas in these bottles. So we are using only one manifold, but here we can have more of the manifolds. So you can flush 6 or 7 bottles in a one go. So then we use this rubber septa to close the bottle. So once the gas is being flushed, after flushing, then we put up the aluminum cap and then seal the bottles. It is like normally you might have seen the injection bottles which are with the rubber septa and the aluminum cap. So this is a sealer through which we maintain that okay, this gas is flushed and we are having the airtight containers. So after sealing these bottles, then we put up these in the autoclave to sterilize the bottles. And after autoclaving, when you take out the bottle, now you see that these bottles are most of the bottles are colorless. Yes. Here you can see that all the bottles have lost their pink color. Only one bottle there was some maybe air entrapped in this bottle. So rest of the bottles are colorless, which indicates that we have maintained the anaerobiasis in these bottles. Then we add the inoculum in these bottles. And for the inoculum, we have already pre-mixed the fecal sample of the human content. And we pooled the sample. Normally it is 4 to 5 to 10 individuals. And then we have to use the syringe to add the inoculum. In these bottles, like 80 ml of content, we add 1 ml of the fecal content in all these bottles. So this is through the syringe. And then again, after adding the inoculum,
Hello. हेलो गुंजन सर, आपकी वॉइस ऑडिबल नहीं है। हेलो गुंजन सर।
Hello.
Yes sir, you are audible.
I'm sorry. I don't know how it got disconnected somewhere. So I don't know exactly where were we. Have you all seen the video?
No sir.
Video?
No sir.
No, nothing on the video.
No sir, last 5 to 10 minutes are left sir.
Okay, but the presentation was okay. Presentation was over.
Yes sir. Yes sir.
Okay, so I will just share the video. So it is about the batch fermentation. Is it visible now?
Yes sir, it is visible.
Okay, so like I said, the batch fermentation is just like very small vessel reaction conditions, and we normally use this.
Sir, sorry, it is paused sir. Please resume that.
Yes, I have paused. I have not started yet.
Okay sir.
Okay, so you can see there is a small glass bottles. We call it as serum bottles with a capacity of 125 ml. And this is the intestinal content which we have a premix of media. And we have already added the feed ingredient or any bioactive ingredient you want to test in these bottles. And this batch system, we optimize first the dose of the active component. So these bottles are filled with like different doses of. This student is working on moringa plant, and we have added different doses of moringa plant extract, and then we add in 125 ml. Normally we add 80 ml of content of this intestinal juice, and after filling these bottles, so normally we add yes. So next stage is that to maintain the anaerobiosis. And this system is which we are having in our laboratory, that is gassing manifold. So this system is connected with three gases: nitrogen, hydrogen, and carbon dioxide. And you can see there are 3 ports, and we can regulate the pressure of these gases through these ports. And this system is having copper wire. Like normally we use this gas cylinder, commercial gas cylinders, but we are not pretty sure that these are free of oxygen. And to maintain the anaerobiosis or to simulate these conditions, we have to be having the strict anaerobic conditions. So this heated copper wire, we first put up the system to heat, and this heated copper wire, it absorbs the traces of oxygen available in these gases. So after heating at 80 degrees for this system, then we start flushing with the gas, and it depends that what kind of mixture you want to have. Either you can have only the carbon dioxide in this system, or you can mix it with the hydrogen, or you can mix with the nitrogen. So this system is manifold system, but we are using only one fold, and we can flush these bottles with the carbon dioxide. And you can see the color of the resazurin is getting pinkish in color in these bottles. So after flushing this gas in the bottle, then we cover it with a rubber septa, and then we cap it with the aluminium. So it is just like of injection vials. Where we have the rubber cap and the aluminium cap, and we seal this thing to maintain the anaerobiosis in the bottles. Is it clear?
Yes sir, it is clear.
Okay. So once you flush the carbon dioxide in these bottles, then we go for the autoclaving of these bottles, because we have to sterilize the media. And after the media sterilization, you can see, except this bottle, all the bottles are colorless. So this greenish color is because of the moringa. Otherwise, all the bottles are colorless, except this one bottle, which means that there is some entrapment of oxygen in this bottle. Then, after completing the crystallization. Then we add the fecal slurry. Fecal slurry is the inoculum in this case. Like in the normally fermentation, we have a pure microbial culture. We can add one culture or two culture, but here we have the mixed microbial composition, and we add this through a syringe. So in 80 ml of content, we add 1 ml of this fecal suspension as a inoculum. So after addition of this fecal inoculum, then because there might be small hole in this rubber receptor, so always that after with that we flush it with the anaerobiasis. So this is the setup of the batch fermentation. How to start with the how to determine the dose of the active component. So after completing this batch fermentation, we incubate, and then after 24 hours, 48 hours, 72 hours, you can take different samples and do further analysis. That is okay with the batch fermentation? Is it fine?
Yes, sir, it is fine.
Okay. So now we move on to continuous system. Video. So once we optimize the dose with this system, then we go for the continuous system that I have shown in the presentation also. With the okay, so this is the continuous system which we are having in our laboratory, and this is the table which I have shown that what are the residence time, flow rate, and pH in these reactor vessels. And these are the peristaltic pumps where all the reactors are connected to one another. This is the circulatory water bath to maintain the temperature of 37 degrees to these reactors. And you can see this reactor, it is small, a very small reactor of 1.5 liter capacity, but normally we add different content like 200 ml, 800 ml, and 600 ml in each reactor. And this is the double jacket vessel with connecting pipe to the circulatory system of the water bath. And on top, you have seen that there are some points where we can maintain the pH. This is the acid port. This is the base port. This is the pH controller port, pH probe. Then we have the feed point, feed inlet, and the feed outlet point. And these vessels are shaken with the help of the magnetic stirrer, small magnetic stirrer. Because this stirrer, it maintains the movement of the content. Like in our body, we have a peristaltic movements. That's because of our intestinal system. But in these conditions, we use magnetic stirrer to mix the reaction content. So these are the 5 bioreactors which are connected in a series, starting from the stomach, small intestine, and the last three are the colon part. These are the pH controllers, where the last part of the 3 column, they are connected with the pH controllers, which maintain the effective pH of 6, 5.5, and the 6.9 in each part of the reactor. This is the acid and the base. So whenever there is a sensing in the pH controller that pH has been changed, accordingly it adds acid or base in that reactor. This is the pH probe, autoclaval probe, you can see there. So whenever there is a change in the pH, it automatically adds acid or base. And this is the pump, pH feed pump, which circulates the feed from one part to the other part. So here we have set the pH of 6.8 and the actual pH is 6.3. So it should add base in this reactor vessel. So in this way, we conduct this series of experiments for 20 continuous days, and like this is the movement of the base. And after the completion of the experiment, then we take, or you can say the different intervals of time, like 1 hour, 1 day, 2 day, 3 day. You can take a sample from this reactor and you can analyze the samples. So this was all about how to set up these simulation systems in the laboratory. I hope it was okay. Any questions or any query?
Yes, Dr. Gunjan, it was very informative. We had this everything, your visualize and your presentation was very good. Any question from the audience?
I think there is no question.
So, Dr. Gunjan, Arjun, these things mostly available what you have added in the different part of the bottles, and you are making the simulated conditions. So you get it from the custom facility, or how you prepare it in the lab?
Which thing?
The component you have mixed.
Yes.
That you purchase from the custom facility, or you make it in the lab?
These are all chemicals. These are all chemicals like we use bile and pancreatic juice, and these are all available chemicals. Okay. The only part we add is the fecal sample.
Okay. So if someone want to test this probiotics or any kind of effect of these microorganisms, then that has to be inoculated in the system and observed.
Yes. The only major you can say advantage of this system is that you have not to go for the in vivo studies. Like for in vivo studies, like for the microbial content, either people go for the fecal content of the mice, or they have to sacrifice the mice. So here we don't have to sacrifice anything. You can take random sampling, you can take many number of samples any time point. So that is the advantage of this system.
Okay. Thank you very much. It was really a great interactive, and it was helpful for the participant actually. The kind of lecture they are listening, actually it was a more of theoretical based. But now they have come to know that it is based on something which can be done outside the system also. Most of the time you are not able to use animals, and there are animal ethical issues. Yes, if we are able to optimize these things in the in vitro condition, that is nothing like that. It's a very fantastic area you are working, and these things needs to be further optimized in the conditions. Yes. So is there any question from the audience? What is this?
How much it is similar to our natural digestive system?
Digestion, you can say, we exactly mimic the digestion, because the digestion happens up to the small intestine. So most of the laboratories in India, they have conducted up to the digestion part, but simulating the fermentation, that is more difficult part. So like what we have seen, that normally during the adaptation period, 50% of microbial population is gone. So that is a disadvantage. But once they are established with the 50% population, then it is stabilized for the period of study.
All right. Yeah. So if there is no question, I really appreciate the kind of efforts Mr. Goel did from his lab. Despite of his schedule, he is having a very busy schedule with that. And I came to know that Mr. Goel is not well also. And despite of that, he managed to join with the GIAN participants. Sir, thank you very much. On the behalf of the organizers, I thank you that you gave enough time and convince our participants.
Thank you, Dr. Sam. I think I have made attempt to make them understand the system.
Oh yes, it was a very informative. Everything was visualized, and people were connected, and they were listening a lot. Thank you very much. Thank you, Dr. Sam. Thank you very much.
Thanks.
So all the participants should wait. We are having a laboratory demonstration after it in our lab in the Department of Genetics here only. So we'll take 5 to 10 minutes to arrange the things, and then we'll connect again. Mutane लिखा है then.
Alright. Alright.
Is it audible? Is it okay? Okay, all right. Welcome back all the participants in the Laboratory of Microbial Genetics in Department of Genetics. Just ensure me, is it audible or not? Is it? Okay, all right. So I would like to introduce some of the activity which is going on in the Laboratory of Microbial Genetics here. My group is working on different aspects of probiotics, its growth and bacteriocin production. There are several projects which are dedicated on isolation of lactic acid bacteria, their growth, and especially the bacteriocin profiling of different samples in the food samples and in the soil samples. In some of the project, we have isolated food isolates of lactic acid bacteria from different fermented foods, like we have dosa, we have curd, and we had jalebi, and other fermented foods were there. And after isolating these lactic acid bacteria from different foods, these strains were characterized for its probiotic property and identified using biochemical and molecular methods. After the identification, these strains have been characterized for bacteriocin productions. So the lab is dedicated to understand the bacteriocin profiling of soil isolates and food isolates, and whether we can develop this bacteriocin as a clinical antibiotics or as a food preservative or different gut microbiota modulation. That study we are pursuing it here. Other than probiotic bacteria, our lab is also dedicated to understand the archaeal bacteriocins. We have Haloarchaea in our lab, which is isolated from salt lakes, and these Haloarchaea are also known to produce certain bacteriocin-like peptide. These are not bacteriocins. Bacteriocins are mostly produced by bacteria, but these are technically called as halocins because they are produced by Haloarchaea. And these Haloarchaea similarly, like they have been identified by using biochemical and molecular methods and considered to be the Halophyrex larsenii. So we have Halophyrex larsenii from the salt lake, which are producing novel halocins, and we have Lactobacillus plantarum and Enterococcus heri from food sample. We have Pediococcus pentosai from the soil sample. One of the strain which was really different than the others were Bacilla confusa, which was also isolated from the soil sample. So the lab has identified certain potential probiotic lactic acid bacteria which are producing bacteriocins and characterizing their genetic and morphological application, structure function analysis against different target strains. So in this session, I would like to introduce you about the typical understanding of initial growth phase, how one strain can be grown and how you can design the experiments, what are the conditions that has to be optimized, or actually the initial parameters. So we use two types of medium in general, like one is, as you can say, for most of the probiotic bacteria, they grow in MRS medium. So till date, what we have, the condition is only a rich medium, nutrient-rich mediums are available, which is able to grow. Till date, there is no minimal medium available for the growth of probiotic bacteria. So whenever you isolate the probiotics from the sample, you have to use a complex medium, a rich media. Only they allow the growth of these lactobacilli or enterococci or other lactic acid bacteria only in the presence of rich medium. One of the medium which is well optimized is called as MRS, and this MRS medium supports the growth of lactobacillus enterococcus only. But there are certain others also they are used like TGY, tryptone glucose extract, and TSY, which has been also explored for the growth of different probiotic lactic acid bacteria. So as you're aware that we are using this MRS broth and the MRS solid agar, and if you have growth on the plate, you have to use agar, and if you are growing in the broth conditions, then you have to use MRS broth or liquid conditions. So there are different advantages of using agar condition. If you see the plate, the colonies are milky white when they grow on the plate. That gives a clear distinction to the other bacteria which are growing on. So if you are using any lactic acid bacteria related to the plate, so if you see how can differentiate this bacteria with the other bacteria, the colony of these bacteria are entire boundaries and milky in color and whitish. So that gives a distinction to the other bacteria which are found. And on the basis of this morphological visualization, one can understand whether it is belonging to lactic acid bacteria or not. So for agar medium, MRS agar we use for Lactobacillus plantarum, and that is used for the growth of colonies on the plate, and there you can maintain it for a short while, not for a longer time. You can maintain in the refrigerator for 4 to 5 days, and if you want to work with this strain for a continuously, that's fine. But if you want to work for a longer time, you have to store it, then you go for minus 20 glycerol stabilization for the long storage conditions. So this is how the solid medium is used for the maintenance of lactic acid bacteria. You can use MRS, you can use TGY, you can use TSY. All rich medium supports the growth of lactic acid bacteria per se, or any kind of, like we are using Lactobacillus enterococcus and Vizela, pediococcus, all are able to grow on the MRS. But these media contains tryptone and peptone, which are very complex in nature, and because of the complexity, they are able to utilize, because the nutritional condition in the gut is also complex. So the medium what you are using is also has to be complex in nature. Till date, there is no discovery of minimal medium for the probiotics, and this is a new where young students can be inspired to prepare a medium for the minimal medium for the lactobacillus, where only use salt. But MRS or TGY, we are using beef extract, we are using tryptone, we are using peptone. All are a complex crude extract of biological cells. So we don't know actually what kind of things are actually required by the organism to grow it in a full extent. So this is another area where one can explore. So this is one condition why we are using MRS and how you can maintain it. The second thing is that you can use the MRS broth. The only difference, we are not adding agar. So you have an MRS broth, and when you use MRS broth, only suppose you want to prepare cells in large number, then you have to use the broth condition. If you want to use cells for your probiotic application in anywhere in the food or anywhere in the system, then you have to grow culture in the broth conditions. Or if you want to go for genetic experiments, then you have to isolate the DNA and the proteins. In that case, one can grow in the broth conditions. The others which helps to understand is the growth kinetics. If you you are using in the growth conditions in the MRS, the really cells are actively dividing and they are able to produce the metabolites which are secreted by these probiotic bacteria. So if you grow in the liquid medium, there are two conditions. Whether you are interested in the cell, because cells perform the functions. So if you grow in 1 liter, grow in fermenter 10 liter, depends on how much you want. So if you want cell for your application, you can grow in broth. Suppose if you are interested for postbiotics, nowadays the next terms has come. The postbiotics are something which is secreted by these probiotics and helpful for the health of the host. So they secrete certain metabolites like bacteriocins, lactic acid, H2O2. What Dr. Chikindas was also suggesting that these are the metabolites which are very beneficial whenever the organism secreting outside while growing. So if you take a MRS medium, you can grow under the optimum conditions and you can remove the cell because bacteriocins per se, if you are talking, because our lab is more dedicated on this molecule. So when bacteriocin is secreted outside, it comes in the medium. It comes on the supernatant part. So centrifugation comes here as a very important part. So why I am emphasizing these two conditions, either it is a agar or it is a broth, because you have to provide a a standard condition. Under what condition these probiotics grow? So if you think of the temperature, most of the time the temperature is like 30 to 37 degrees centigrade. Most of the probiotic bacteria, they grow at the body temperature, which we have, because it's a real application is in the human host or animal host. So the 37 degrees centigrade is the body temperature. This is the reason I found whatever we are working is mostly they optimally grow at 37. But there are reports which like at 30 degree these probiotics are able to grow at the 30 degrees centigrade also. That's fine. चल तो रहा था, क्या करती? It's fine. I'm sorry, it was some disturbances. So we are talking about this MRS medium and the growth condition of these probiotics. And whenever you grow, you need certain conditions. Number one, the temperature. Most of the probiotic bacteria, they require 37 degrees centigrade temperature, which is a body temperature. But there are certain reports we have found that 30 degrees is also favorable for the growth. So they grow near 30 to 37 degrees centigrade. One can put the temperature in the incubator shaker, the incubator at the 37 degrees centigrade. Now, the second most important condition is the pH of the medium. These strains, they happy to grow at pH near neutral. It's not at the exactly pH 7, but the MRS pH is, for example, at 6.8. You can use this 6.8 pH of MRS and put it 37 degrees centigrade, inoculate your strain, and will be able to grow. What about the aerobic conditions? Most of the lactic acid bacteria are facultative anaerobes. Means they are aerobic. They are also, but they can grow in the anaerobic conditions also. So if you provide the shaking condition and don't provide shaking condition, that doesn't make much difference in the growth. So mostly if you inoculate and put under static conditions, these strains are happily growing, so you don't need shaking condition for the growth as such. But for different experiments, you can plan to shake if you are using some kinetics or if you are using some statistical analysis, that can be done. But just for the growth of the cell, it it is only that you can put it here and the 37 degree setting it is sufficient. So 37 temperature, pH is 6.8, and static conditions are favorable for the growth of probiotic bacteria. Incubation period we have seen that differ from 16 hours to 24 hours or 30 hours maximum. So within an overnight 24 hours is sufficient for the growth of these bacteria in the laboratory conditions that can be done. So we are using one system like for the growth in the broth conditions and growth on the solid media for the maintenance. And why these broth conditions are grown, that was I was discussing. I think it was interrupted, but I'm just repeating the things that you are using broth conditions for the collection of cell. You can utilize the cell for different purposes, for applications, or if you are using growth kinetic experiments, you can grow in the broth. The second most advantage of using in the broth condition is that you can collect your self-free supernatant. So when you collect your self-free supernatant, most of the secretome, everything which is secreted by the probiotic has come in the self-free supernatant. And for example, we are using bacteriocins. So it comes in the supernatant. You take the supernatant and collect it. And after the collection, you can test the presence of this antimicrobial compound, whether it is present or not. You can test against the indicator strains. So if you have any indicator strain Micrococcus luteus, you can grow the Micrococcus luteus and find on the plate, that's fine. So one can take any specific indicator which is sensitive to those, which yesterday we were discussing. So this is a plate which is a very good yellow color colony. It looks like over it, if you can see, very good yellow color colony is visible. So we are using in our lab Micrococcus luteus as an indicator strain. Why Micrococcus luteus? Because it is one of the most sensitive strain, and number two, it gives a very good yellow color. Why yellow color is important? Because it creates a contrast while visualizing the zone of inhibition. So what we have is here, if you see this plate like this, it creates a clear zone. And when this yellow color is something like a Micrococcus, and the zone, if you see, put the CFS in the well, it creates a fantastic zone of inhibition, which indicates that the probiotic is secreting certain antimicrobial compounds. So whatever it is, you have to identify that these compounds are lactic acid, or these compounds are bacteriocins, or H2O2, that has to be identified. So in the purification part, we are going to have a different session, but I'm not going to disclose you today. But in the separate session, we will have how these compounds from the CFS can be purified to the homogeneity and can be tested. So you can determine whether CFS contains your bacteriocins or other compounds or not. So today's tutorial is more based on the growth condition of these probiotics, and while growing, what are the precautions has to be taken in the medium conditions and others. So our laboratory is dedicated to understand two conditions. One is in the liquid, another we have started on the solid state fermentation. Solid state fermentation is something where you can do the probiotics on the inert matrices or agricultural waste matrices one can take, and this is a very good advantage for the utilization of agricultural waste. Nowadays, lots of agricultural waste are available, because earlier time it was recycled, but nowadays no one is able to recycle it completely as it was happened earlier. So these probiotics we have seen, they are able to grow on the agriculture waste, like we have seen on the wheat bran, we have seen on the rice bran, oil cake. So they are able to grow over it. So what is there that if you take this like is wheat bran, is put it in the flask, and this is solid SSF experiment, solid fermentation. That if you take this your probiotic and inoculate over it, and they are able to grow and utilize this wheat bran also. So, what is the advantage that one can, that why you are growing in the SSF condition using the matrix? And if the answer should be that most of the probiotics strains, they grow on the intestinal surface. So, which real application is in the solid conditions. That most of the probiotic in the gut, they grow in the epithelium of the human and perform the function. How? Because most of the probiotic cells are having ability to adhere with the surface, ability to adsorb the surface. That is a hydrophobic behavior of a probiotic cell. And the probiotic, most of the probiotics, they are hydrophobic in nature. The hydrophobic behavior is considered to be one of the important probiotic property. And if a cell doesn't has this kind of affinity to adsorb with the matrices, that it will not be able to perform the probiotic function. For example, if you take a probiotic cell and it goes in the intestine, and if it is not able to adsorb to the epithelial cell, then it will not be able to perform the function. So it will go and pass it. So no, the cells has to has an adhering property. If it adhere better, and this one is something we are going to perform a separate experiment to demonstrate you that what are the hydrophobicity experiments one can perform in the laboratory. But today we are just trying to show you that there are some solid surfaces can be used, like wheat bran, rice bran, and all cake can be used for the growth of these bacteria, probiotic bacteria. We inoculate this probiotic bacteria over it and incubate at 37 degrees centigrade and see its growth. So definitely in this condition one cannot take the photometric growth because it's growing in the solid surface. So you have to calculate the colony forming unit, the CFU. Whereas in the broth condition, whenever you grow like that, then you can take the absorbance, you can take the OD. But here it's only you can count the CFU per ml. And of course you can wash this matrices, and after the washing you will collect the cell-free supernatant and wash medium with a buffer that is produced almost the secretory compounds. Whatever you have, which are lactic acid or bacteriocins or anything which is secreted by acetyl, anything which is secreted by these probiotics has come in the buffer. And you can collect this buffer and test whether bacterial production is there or not, or any metabolite production is there or not. So, in broth condition, you are going to understand the laboratory process and growth kinetics, but the real application of probiotic is the solid surface. This is the reason we are optimizing this condition under the SSF condition, and therefore this can be applied for different applications. And second is that these are agricultural based residue and much cheaper than the commercial media which are available in the market, like MRS or TGY. They are very expensive in nature. So this provides a kind of cheap material from the agriculture waste which can be recycled, and there will be a clear polarization of this product that you are preparing a product, making a probiotic out of something which is a waste that these probiotics are able to grow it over it. And whenever you inoculate it, you will find the growth, and the growth condition can be stayed in terms of CFU, right? Of course, you have to go for optimization also. The condition, either you are growing in the broth or growing in the SSF, the condition has to be optimized. Whether what conditions in terms of nutrient media, the nutrients which are available, or you can have a temperature, you can have a pH, or you can have a matrices which are available. So that also needs to be optimized, because there is a difference in the growth kinetics in the broth and the solid surface. If you grow the things on the solid surface, the growth kinetics is entirely different than the broth. Right. So that has to be optimized in terms of its number of cells, amount of metabolites secreted. In our lab, that we are also focusing to understand that what are the optimum conditions should be there for bacteriocin production. Mainly our lab is dedicated on the bacteriocin profiling productions, how it can be enhanced for the large amount. So there are different steps one can do for the optimizing the condition. One is one variable at a time, though it is a older technique, but at a student level, one can start by optimizing one variable. You can change the different variables one by one at a time and see the output or production of bacteriocin which is taking place, or growth of probiotic which is taking place or not. Second, one can go for a statistical optimization, which is a very reliable tool, blanket Berman design and response surface methodology, RSM. So both are very much have been used for the optimization of different microbial condition or any condition in the biotechnology. One can optimize using these parameters, and we are using this blanket Berman design to understand the important factors which are responsible for the growth. You can select what factors out of the 10 factors you are using, which one is important. You can use blanket Berman design to select one or two factors which are important, and then by selecting one, two, or three factor out of it, you can do the response surface methodology using CCD design. And this CCD design does not only see the level of variation but also interaction of two different factors. So you can see the interaction of one factor like glucose with the fructose, or nitrogenous sources, or peptone with the yeast extract, any media component that can be used, or if you have different variables that can be used for response surface methodology. And there it gives you a clear idea that which factor is interacting for enhanced growth, which factor is interacting for the higher production of specific metabolites. So this is what we wanted to demonstrate you, that probiotics, whenever you isolate, these are the conditions had to be optimized in the laboratory, which is a very basic, and but it is important to understand the growth, because if you are using a cell, you have to grow in a live conditions. The cells are completely mature and live, only then they will perform the probiotic function. And if they are growing under stress and they are not able to completely divided or multiplied, or growth kinetics is not completely followed, then they will not be possibly able to perform the function. Right. So this is the reason that though it seems a smaller experiment for the student, but I appreciate for them motivate to grow the culture in a best possible way, best possible manner, so that you have a live, active bacterial cells which can be used for their application, and they produce these metabolites only in a condition where when they are live, when they are active. If they are growing under a stress condition, they are not able to produce these metabolites in an optimum level. So the growth conditions, right from the media, right from the temperature, right from the incubation conditions, what you are using, is very, very critical and important. What problem we are facing, we already know that in metagenomic studies suggest that only 2 to 3% are culturable, or 1% hardly even culture. So if you take a sample, you don't expect that all the microbes which are in the sample will be able to grow, because we don't know the exact nutrition for all the microbes which are present in the soil sample or food sample, whatever it is. And whatever the medium we are providing, for example, if you are providing MRS, that is suitable for one set of microorganism, but it may not be suitable for other microorganisms. So that is one bottleneck we are suffering to understand, that how most of the microorganisms will be able to grow on the plate. So maybe in the future, in next generation, that certain media are made in such a way that most of the microorganisms which are present in the sample will be able to grow. But till date, we don't have that bottleneck is facing. And the problem is being solved with the metagenomics. What Dr. Chikandas yesterday was saying, that the metagenomic approach with the probiotics is very important to know the function of those probiotics which are not able to grow on the culture medium. So, I mean, we should learn those technology also, which allows the growth of other probiotic bacteria, which we don't have medium for them. But they perform the very important function in the gut. So, we know very fraction of bacteria which are able to grow under certain conditions, but they are majority of them, they are not able to grow. So, this is what I wanted to tell you in this today's tutorial. Anything, if you want to interact, I will come on the computer and tell you. Okay, dear participant, do you have anything to interact or any question? Then we can discuss it. don't see any question related with my presentation in the chat box. No? Okay. You don't have anything? Alright, if you don't have any question, then we can stop this tutorial here only, and we'll see you in the evening. We'll meet in the evening at 5:30, right? See you.